Okay, I think we start. It's my great pleasure to introduce Alejandro Trujillo. We've worked together at JSU and kept in touch since then, and we're working on some projects together, collaborations, and it's a great pleasure that he was able to come here to OIST and join us for a couple of weeks. He's now a senior scientist at Canberra University, and we'll be talking about his great work he's doing in eDNA and biosecurity. Take it away.
Better? Okay. I'm just going to take this off then. I'd also like to acknowledge that all of the work that I'm going to be talking about today was funded by the Department of Agriculture, Fisheries and Forestries in Australia. They have funded most of the work that I will be talking about today. And I want to recognize that it's thanks to them and thanks to amazing champions at different levels of government agencies and state officials that this research that I'm going to be talking about is having the impact that it's having in Australia. And then, of course, this wouldn't be anything without the absolute cornerstones and rainmakers that analyzed the samples, collected the samples. This is my team in Australia. I'm very happy to present their work and the things that they have been able to produce. And without further ado, I'll just start by giving a brief intro of what it is that I do. I'm in reality an applied scientist. I try to get things that I know we can do in the lab, and I apply them into field applications and see where they remain suitable and trustworthy when used for practical applications. In the case of biosecurity, biosecurity is nothing more than just a set of measures, of protocols that we have available to us that we implement so that we can protect our primary resources, our environments, and our livelihoods. It is something that continues to be ever complex. It is mostly affected by trade and movement of people, but as connectivity between countries, as technology gets better, then pest translocation between countries, continents becomes worse. In the case of Australia, for example, it's been predicted that by 2025, there's going to be a significant increase of tourism, of people coming into the country, as well as containerized cargo, with bringing in quite a lot of commodities and other primary resources from other countries. And this, of course, presents quite a big risk for Australian livelihoods. And this is the reason why Australian biosecurity tries to modernize itself. It tries to find and adapt better ways that we have available to us so that we can minimize the translocation of pests, detect these pests when they come into the country, and have a better opportunity to mitigate their impact if they were ever to become introduced within Australia. Now, within that context, there's many, many techniques that we have available for us. And many of the techniques that people try to implement are those that are quick, that don't involve quite a lot of effort in sampling regimes. And it brings me back to one of my favorite examples from old school ecology. This is Professor Terry Irwin, who unfortunately passed away in 2020, but he invented a technique called tree fogging. And tree fogging was essentially a method where he was trying to assess the diversity of insects that were inhabiting a given ecosystem. And the way that the technique that he created was essentially grab a massive amount of pesticides and just fog one single tree in the rainforest. And then all of those insects would fall. He would capture them all, and then he would assess productivity based on the amount of insects and species that he would find. We have, of course, moved a long way since the 80s, but eDNA remains quite within that concept, where we're trying to find these proxy methods to assess diversity, to measure and quantify the species that would occupy a given ecosystem. So eDNA is quite simply DNA that is left behind in a given environment. Now, if you like definitions, this is one of the definitions that I like the most, which is DNA that is collected from an environmental sample without an attempt to isolate the actual target organism that you're looking for. Yes, you may capture bacteria and viruses and so on, microscopic organisms, if that's what you're looking for. But the main concept is that you're looking for a method, a proxy method, that would inform you on the activity and presence of your target animal without actually trying to grab your target animal. And within that context, eDNA has seen an explosion of applications, of research, of innovation, because it quite reliably can tell people what kind of organisms can occupy a given ecosystem. And within that, there's been a lot of research looking at which mediums, which matrices are the best ones to capture eDNA, and how we can process those mediums. So eDNA in itself is not a technique. eDNA is just another sample for which we have many techniques to collect them, to extract them, and to analyze them. In biosecurity, most of the, let's say, attention has been driven by marine science and biosecurity applications in the marine system. Many of these applications have already been reviewed. They talk about the limitations and some of the benefits that we have available for us, but very few have started seeing their applications in the terrestrial system and across the biosecurity continuum, which is where I work. It is a very different system where I apply my science. This is a picture right outside of OIST, right next to the beach, and this is mostly a representation of where most of the research that has been done within the context of biosecurity has occurred, which is the marine system. There's many limitations with this. People that work in this room with eDNA are already thinking, well, sampling effort, degradation factors, inhibition, contaminants, and so on. How big is the ecosystem, and how can we collect a sample that is representative of the ecosystem that we're going to work on? This is very different from those, let's say, environments within which I work. Many of the applications that I work with are not a huge environment. They're much more, I guess, in a lower scale. There may be just packages and bags full of animals, or they could just be shipping containers within which there's a defined space, a defined area that you can sample and get an information based on eDNA or RNA later on. I work across the whole biosecurity continuum with my applications, which made it a bit of a struggle to know what I wanted to speak about today, because I work with quite a lot of organisms across the biosecurity continuum. I'm not going to be able to speak to you all about every single one of them, but I am keen to have further conversations if you're keen to know more. Today, I think I'm going to concentrate in two different case studies, which is, one of them is the ornamental trade, and the other one is the case of shipping containers in Australia. So before I begin, just diving into this whole thing, do me a favor, if you have any questions, just to stop me, raise your hand, let me know what you think. And if I start getting like overly excited and speaking really, really quickly, just to stop me as well, because I have a tendency to get overly excited when I talk about the things that I like. So in the case of the ornamental trade, this is probably one of those pathways within which eDNA applications are the most, I guess, straightforward. You're not dealing with a marine system, but you're still dealing within an aquatic system within which samples can be collected quite reliably. It's comprised of course, freshwater and marine species, of which most of the species that are traded are farm-bred and freshwater, while the remaining ones are from the marine ecosystem, of which about 90% remain to be extracted from the wild. Now, there's two problems that we have with the ornamental fish trade. The main one is the case of non-permitted fish within the context of legislation and regulation of imports. So a non-permitted fish species is that a country doesn't allow to be imported into the country, right? Allowing for the redundancy. And this is one of those perfect cases. One of them is permitted, the other one isn't. You can see a few differences, right? And you can see like the fins are different. You can see some coloration that is different. The spotting pattern is different. But imagine this in a formal application of biosecurity where you're an officer, a bag full of these fish arrives, and you need to have a look at them. and essentially say, in the question of minutes, do we have a bag with non-permitted fish? It's quite impossible. Like, even if you have years and years of training, being able to see in a bag through the noise of those species that are indeed permitted and find that one that is not permitted is very difficult. The other problem is the case of microscopic infestations and infections. So this is the case of two parasites that I used to work with during the time of my PhD. One of them is Argillus japonicus, and the other one is Dactylodryas vastator. Dactylodryas is a gill parasite that infests fish. And those very commonly come undetected at border control, quite simply because how exactly are you going to be able to detect them if they have low infestation numbers? These infestations would have to be quite clear and obvious for officers to find them. So one of the main things that I wanted to do at that stage was to assess, well, could we use eDNA in offshore applications to inform officers on the presence of non-compliance? Essentially, are fish that are coming into the country that are illegal, and can we detect parasites? And the two methods that we selected at that stage were, well, for the purpose of fish, we're going to be using HTS, so high throughput sequencing and metabarcoding approaches using portable technologies. And the other one, for the detection of parasites, we're just going to be using the species-specific detection using qPCR. There are portable technologies that can do this, but all of these portable technologies do have their own little, let's say, minutiae to them. Like, there's little things here and there that you need to fully understand within the operational context that complicates the way that we can use them. So we went to Thailand, we went to an international wholesaler, we collected water samples from their quarantine facility. And within this quarantine facility, we took water from the intake. This is water that is coming into the facility. The packaging water, this is water that they actively use to pack all the fish and send them over to whichever country is bringing in the fish. In the case of Australia, Australia requires that fish that are quarantined are remained in a self-isolated system just for the purpose of importation to Australia. So we collected water from that. Then the international quarantine, which had pretty much everything else, and then the wastewater, the outlet. In the case of Australian quarantine, we knew that in the tank where we collected the water samples, there was this one species of fish, which is the red-tailed shark. It's not actually a shark, but it's just a colloquial name. And then in the other one that was importing fish internationally, there were goldfish, but that system was connected to many other tanks that had multiple other species. So we were expecting to see a couple of important topics that I'm going to be discussing later on. The main one being to answer a question that we actively had from biosecurity officers, and is that how quick can we do this? Like, how much time are we going to lose if we actually implement these technologies within our daily work? Keep in mind that by that time, there was no regulation, no protocols that would indicate how eDNA technologies could be used in the daily life of a biosecurity officer. So the question quite simply was, what exactly can we skip so that we can still retain a viable answer within the minimal amount of time? And within that context, I started thinking about, well, what do my stakeholders need? What is the end officer looking for? Can they sample water directly? Can we just take water directly, no processing, and can we actually analyze free-floating eDNA, right? But that was a problem, because when you have uncleaned water samples, there's quite a lot of inhibition and a lot of contaminants that are just floating around in the water. They're clearly going to inhibit your sample. So these are the outputs of quality that you have when you're using Oxford Nanopore Technologies mean ions. I believe you have one downstairs in one of your labs. And what you can see is the standard on the top. That is kind of what you're looking for in terms of quality. But then below, you see the quality that you get from samples that are PCR-free and in clean environment, and then below, PCR-based. So they're being amplified from dirty water. In both cases, there's a minimal amount of sequences of quality that you could actually use for the analysis that you're going to be doing later on, which is unreliable. But if you purify those samples, and then you continue your approach, then quality immediately jumps up. And that is something that you need to be clearly stating to your stakeholders, that there's no way to actually cut corners within the workflow that you need to establish if these were to be used for biosecurity. We also compared the portable technology against the, I guess, let's say the gold standard that we would normally use in the lab setting. Of course, the one in the lab setting has quite a lot of reads of quality that we could use for inference across 3 different genetic markers, which is 12S, 16S, and COI. And in the case of the mean ion, we use 2 different, let's say, kits for library preparation. And across all of those 3 markers, you see that there's a reduced amount of sequences compared to the gold standard. Is that a breaking point? Not exactly. I mean, you may still have sequences of quality for you to inform your officers on what you're getting. So these are the results that you get from all the samples when you're using, in this case, 12S, the 12S genetic marker, using nanopore technologies. The first thing that you see is, of course, quarantine. The international quarantine sample just has a massive amount of sequences, followed by wastewater, and then in the case of Australian quarantine, just a small amount. What is interesting is that in both instances, we're able to detect the fish that is actively in the tank. Of course, in the case of international quarantine, there's a much larger number of species that are, for which eDNA is being captured. Of course, it's a recirculating system that is sharing this water across all of those species, which is not the case for Australian quarantine. And the other really interesting thing that was done at that stage was that we could see that what we consider clean water within the context of eDNA was indeed actually relatively clean. There's no traces of other organisms that could provide background noise if these fish were being imported into Australia that could limit the amount of information that you could get out of them. And this was interesting because from one of my PhD papers within which Roger was actually a co-author before, we demonstrated that dirty water does indeed increase the amount of false positive detections that you can get within a biosecurity context. False positive detections are a big problem within the context of biosecurity, and it's something that you're actively trying to minimize. And what is being done at the moment is that clean water has that definition of, well, what exactly is clean? Are we looking just for clean water in terms of no contaminants and so on, or are we also thinking about the context of eDNA and traces of DNA that could be in the water? In the case of species-specific detection of parasites, this was much more straightforward than what it was using the mean ion technology in the field. So that thermocycler that you see in the screen, that's a Franklin thermocycler from a company called Biomine. It is very simple to use, to the point that the owner and the manager of the facility were the ones that undertook this analysis when we were doing this over there in their facilities. And one of the interesting things that we were able to demonstrate is the capacity of RNA to act as a baseline of detection for metabolically active cellular activity. Essentially, are organisms alive when they're shedding this RNA? So in that sense, eDNA and RNA have received a lot of attention, but RNA remains to be indicative of active cellular activity. And we could demonstrate this from samples collected at the quarantine facility. So we went straight to fish that were treated for against infections of Dactylogyarus, and fish that were not treated for infestations of Dactylogyarus. And what you see in the screen is the signal of amplification that you get from our positive control. This is just genomic DNA of the parasite. The DNA, the signal that you get from non-treated goldfish, these are goldfish that are infested. And then the signal that you get from treated goldfish, goldfish that in theory should be clean of the infestation, but you're still seeing detection, right? This is what you see when you're analyzing RNA. So you see your positive control, and the only signal that you get are those from non-treated goldfish. So it showed to government that you could actually use this technology to inform your officers on compliance of health requirements. Could you use eDNA to make sure that those fish are indeed not infested? So it triggers quite a lot of research. It triggers quite a lot of higher value researchers that can be using these applications. And some of the outcomes that we got from the ornamental trade being quite complete is that as an officer, you need to first reliably use the workflow to clean your samples. When it comes to using high throughput sequencing technologies, it is not that straightforward, especially because in the case of Oxford Nanopore Technologies, all of those consumables need to remain within a cold chain operation. So they need to be kept cold, essentially. And that is not always possible when you're out in the field. In the case of biomine consumables, the shelf life is much longer and it can remain at room temperature. So these are all considerations that one must have when you're telling your officers how this technology can be used and how it can be applied. So quite a lot of lessons that we got from the ornamental trade. And many of these things we took to a much bigger application that I'm going to be talking about now, which is the case of capra beetle. So this beetle is one of the worst pests of plants, dried products in the world. It's a species that is known to infest about 170 different dried products like seeds, like rice, like nuts, and so on. It is also known to infest oil products, animal products such as leather, and it has a very quick active life cycle within which the life cycle can be completed even within one single commodity, and it can just reinfest and reinfest and reinfest one single stage. They also can remain dormant if conditions are not appropriate for them to close their life cycle for years on end. And they can just find a very small crevice somewhere and stay there for a matter of years until those conditions remain to be viable for them, and then they close the life cycle. And they bring in a really big problem because when it comes to detecting them, they're quite small. They're about 3 millimeters long, right? They're quite tiny. And their infestations only become evident when there's quite a lot of them, right? When the whole thing has been infested, or when you have officers that are highly trained in the capacity of knowing where to find them. They're quite overly dispersed in the north of Africa and the Middle East, but because of trade and those connections, and most of the ports, some of the bigger ports in the world are right smack in the middle there. So containers get infested with these beetles. Commodities can get contaminated from those ports of origins. And Australia, in the case of Australia, we're starting to see incursions of this beetle coming into the country, which was the case of 2020. So in 2020, there were a few detections of these beetles coming into the country inside, of all things, refrigerators, right? So a commodity that people wouldn't normally think about at that stage. And when they went to find those infested commodities, they realized that it was also the cupboard that was used to pack those refrigerators that was infested with the beetle. That stage, I had already finished up the work with ornamental fish. Government had already an indication of eDNA. There's something interesting here. So how about we try to test this in the case of capra? So as soon as I heard that there had been these detections, I just grabbed my bags of samples and things, went over there, and then myself went inside, collected dust samples. And the reason why we collected dust samples is because that's the one medium that officers collect when they're doing these detections. So they actively take a vacuum cleaner or how do you call it? A dust, yeah, like a dustpan. And they collect bags and bags of dust. They put them into plastic bags, and then they have to sieve through all of them and find these minute, tiny little beetles. So in my mind, I was thinking, well, if that's what they do, that's the medium. How can I apply eDNA-based detection technologies to that one medium? You don't want to change much of what officers already do. You want to make them complementary to what they can do. So I collected dust samples directly from the bag that they were using, but I also took my own vacuum cleaners with filters and a couple of other mediums to see if I could actually collect airborne eDNA. Could be the possibility. And at that stage, I could demonstrate that, yes, I could actually extract eDNA from the beetle, from this debris and dust medium. And I could also use portable technologies within the facility where this biosecurity response was being done, and I could detect capra beetle eDNA. This happened in 2 occasions, right? On the second occasion, I had a problem. Or more than a problem, was more of a quick revelation. And I started noticing that the officers had that same vacuum cleaner that had been used in other applications. So I asked them, well, has this vacuum cleaner been used in other biosecurity responses for capra beetle? And the quick answer was, well, yeah. you know? Like officers at that stage, their priority was not to collect reliable samples for eDNA testing. Their priority was to destroy the beetle, find it and eradicate it. So they just needed to collect that one sample. And when I published this paper, one of the main things that I made it clear was that for eDNA applications to be reliable within this context, we needed to find new collection methods that would isolate and minimize the possibility of false positive detections. Essentially, how can we cleanly collect a sample from one location to the next? That triggered quite a lot. Oh, this is just what I just said. But this triggered a much bigger project in biosecurity. Government was very interested in the results that we had because it was a reliable method that would detect Capra beetle in the fraction of the time that officers needed to sieve through all of this dust, right? So they provided funds to go to the Port of Brisbane, which is the screen that you see on the top, and we collected these samples, these dust samples from about 2017 shipping containers for between May and August, right? And then at that stage, I already thought, okay, how can we modernize the methods that we use to collect this dust sample? There's no novelty in that technology. That's just a handheld vacuum cleaner with something that they call a dust interceptor. And it's just like a little canister that pretty much just captures all the dust and muck and debris. But by doing that, then I reduced the incidence of false positive detections between samples and between the collection factors. So that allowed me to reliably analyze each one of the samples with the understanding that there was not going to be any cross-contamination between the samples that were being collected. And at that stage, we had already optimized the assays so that they would be able to detect eDNA and RNA of Capra beetle. This medium brings a lot of issues. We may not encounter like the massive ocean, the bigger ecosystems within which marine biosecurity is commonly done, but we encounter very similar problems. And this one highlights one of those issues, and is that the medium itself is quite diverse. These shipping containers move anything and everything between countries. And because they do that, then the amount of samples and the composition of those samples change dramatically between containers. You can get something clean, which is what the things that you see to the left, which is being used to move, for example, food products for human consumption. But you could also get the containers to the right, which is used to move fertilizers, right? All of these leave traces behind, and those traces are contaminants, inhibiting factors, a lot of problems that minimize the reactions that we commonly get in molecular technologies. So what did we have to do at that stage? Well, we wrote protocols. We wrote high quality protocols that would allow not just the scientists like myself, but end users and other officers to understand how to develop protocols for sample collection, how to minimize false negatives and false positives as well, and how we could reliably eliminate inhibition and contaminants when undertaking these reactions. So we used these baselines to analyze all of these samples. We purified all the samples from the shipping containers, and we also expanded the range of species that we were detecting. So at this point, it wasn't just Capra beetle. We also were looking for brown marmorated stink bug. We were looking for invasive ants, in this case, an electric ant, and then the gypsy moth. All of them are quite important biosecurity pests, not just in Australia, but globally as well. And we developed assays for all of them. In many cases, we try to use an assay that would be applicable for both eDNA and RNA together. In other cases, that couldn't be possible, so we had to just develop an assay that was suitable for eDNA and another one that was suitable for RNA. And these are the results. And what bothers me is that we have a problem in science because we present this data in these tables and it never makes it, like, it never gives it justice to how much work actually went into making that tiny table. Like, this was the product of months of collaborations, of brokering agreements, of going out to the shipping containers, looking ahead for these massive tractors that would just move containers all around you, making sure that the samples were properly collected, making sure that the samples were properly purified and quantified, making sure that the assays were working properly. And it brings me to that tiny table. And at the end, this is what government is going to use to understand the incidence rate of this pest coming into the country. So although it looks tiny, it is a table that has the effort of quite a lot of people and a lot of time ensuring quality and control. I don't know if you all get the same problem, but like this certainly is a problem that we need to change in our publications. There should be like a little thing that says how much time was taken to do the whole thing. In any case, sorry, I digress. There were two things that came out of this study, two very important things within the context of eDNA. The first one was that eDNA and RNA detection was shown to be complementary to normal biosecurity inspections by actual officers. This was something that happened. So what you see on the screen to the left is a biosecurity officer that at the time detected one capra beetle from a shipping container. They didn't tell me, but what they told him is use this technique to collect dust samples and give it to us. This was outside of the whole test and all the trials that we were doing. They sent me those samples blindly. I had no clue that those samples were coming. And when I had those detections in the lab, the first thing that you get is fear, like you're freaking out because you're getting actual detections for the first time of a very important pest. So I called government, told them, hey, we have this positive detections in one of your shipping containers. This is important. I think I advise you to trigger a response. And they said, oh yeah, we know. We've already collected the samples. We already know that the capra beetles were there, but it's good to know that the techniques are working. So that's one thing that came out of that. In which they were able to anchor down my results to a confirmed positive biosecurity response in the field. So victory number one. And the second victory that we had is that after that, we started testing all of the shipping containers. Two of them came back positive across the board for eDNA and RNA and were confirmed positive by Sanger sequencing and so on. So I triggered an advice for government. We recommend that you go back and look at these two shipping containers. And they did. That is the first time that ever happens within eDNA in Australia, at least, maybe the world. But when they did that, and they went back to check on those shipping containers and the commodities that we found inside, they found live adults, they found larvae in those containerized cargo. And then that triggered an active biosecurity response in Brisbane, which is probably the first time that ever happens for the purpose of eDNA. So victory number two, because we're able to show that eDNA can be reliable, we can depend on the detection of eDNA. But it also brought together a couple of other problems, and is that happened in two containers out of 2017. That doesn't indicate that the other 15 containers were not infested. It just means that I wasn't quick enough to provide those responses to government. So we need to optimize the way that we collect, process, and analyze our samples for the purpose of biosecurity because things happen very quickly. And in the case of my lab, we could have a return of maybe 14 days, depending on Sanger sequencing and how long it took to produce these sequences and so on. But government needs something quicker. And that is going to be a complication for quite a lot of labs, including mine. So that is certainly something that needs to be understood. But even then, we get to this point. One day we had a meeting with government and we sat down, we started seeing all of these slides that they had produced, and then this one comes up. And I had never seen this before. So I asked them, well, what is this? And they said, well, this is your work. So this is the approach rate of all of these pests. This is how many you found in these shipping containers. This is how many we found for eRNA. This is the number of container counts of connections between containers that you found as being positive versus those that weren't found positive. But these are all the possible connections that we can find. They could even tell me from which countries these containers came from, which countries those containers stopped, either unloaded and then reloaded again. Like there were just a massive amount of information that they could do just on that one figure. And that was the first time that I saw, oh, my work is actually being used. Like that never had happened to me before. So I'm quite proud of this little slide because it shows how government was using species-specific detections of eDNA and RNA for active analytical purposes within the federal government. So that was really, really cool. How are we going? You're all doing okay? Yeah, okay. Now, the second part of what we wanted to do was assess insect diversity. And this is one of the main things that I've been doing here at OIST with Roger. We are analyzing all of this data from all of these containers, but instead of using those species-specific assays, we're using broad-spectrum primers that just amplify insect diversity. And by amplifying insect diversity, they just amplify a lot of non-target other groups. We know that it's a thing that happens, but for that purpose, we used a very heavy sequencing approach that provided about 6 million sequence reads per sample. So far, we've analyzed 951 of those samples. And then one of the things that Roger was able to create, this is why he's amazing, is that he, what is it that I had to do? He created this screen, sorry, I'm just going to change screens for a second. This outline, and I kind of hope that some of my stakeholders from Australia are sitting online and looking at this, because this is what can be done with metabarcoding applications in biosecurity. So these are all the samples that are being amplified from a range of 94 samples. You could go into Metazoa, and then that closes down some of the species that are being amplified, Protostomia, and then you reach arthropods, right? And then in arthropods, you have a whole diversity of animals that are being amplified, including arachnids, including crustaceans as well. But if you were looked for just the insects, then you find an even bigger diversity of organisms that are coming in through these shipping containers. Now, this is just an initial view of what can be done, and we're still processing these samples to assess like species level ID. But it certainly shows the capacity of what could be done, the diversity that could be understood. And one of the things that I want to do at the moment is take this data, because we've of course only analyzed eDNA, but what if we could analyze eRNA as well, and just make that comparison of what is being detected with eDNA and what is being detected with eRNA. So certainly one of those things that came out of this visit. Okay. Why does this all matter? Like, why do we do biosecurity? Like, why do we try to enhance detection of these pest priority species? Well, I'm going to tell you a real story that is currently happening in Australia. So what you see there is a bee, and that red spot that you see on the left, that's a mite. It's a parasite. The genus is Varroa, and that one species is Varroa destructor. Is one, if not the most important pest species of honeybees all around the world. It is currently not established in Australia, but in June of this year, there was an incursion that was detected. And with that incursion, Australia had to trigger a quite large biosecurity response. They started to destroy infested hives and those non-infested hives that were in close proximity to these ones. So quite a lot of hives were being destroyed. And when it all began, that's on the 22nd of June. And that's the port of Newcastle in the New South Wales, like eastern state. And this is where we are now. Well, not now. This is actually from the 11th of August. So in just that small period of time, there were a couple more incursions and detections that were made across New South Wales. And it's a dire situation. Quite simply because it is not just the number, it's the livelihoods of a lot of people in that state, is what they have every day to live. And if that doesn't trigger a degree of empathy, it's something that needs to start happening when you're a researcher working with biosecurity. You're not just doing this for the sake of research and positive versus negative detection. What you produce may become something that in the end is going to help many, many people later on. And that is exactly what we're trying to do with eDNA. And we've had amazing champions of this technology. And we're now working with Agriculture Victoria, the Department of Agriculture, Fisheries and Forestry, as well as stakeholders in New Zealand, to see if we can optimize assays to detect varroamide. Right now, we are able to detect varroamide eDNA using just swab samples as you would like for COVID testing, right? So we just swabbed the entrance of a hive. I actually had a video, but my computer sucks and I couldn't actually make the video work. But what you see there is a biosecurity officer just swabbing the entrance of a hive. And only that swabbing, very quick, reliably told us if there was any DNA detection on debris, on honey, and other dust that just accumulates in commercial hive, for example. So we're currently doing this work as we speak. And I'm keen to see what else we can produce out of this that can help biosecurity officers produce something, like protect some of these livelihoods. So what else comes next? Like, what else do we have in the pipeline? We're also going to be working with yellow crazy ants and red imported fire ants. These are two very important pests globally. In Australia, they have, each one of them has an eradication program. Yellow crazy ants are mostly established in a part of Queensland, within like Cairns and Townsville, which are two different cities in the north, for which there is about $56 million worth of funds to eradicate species. In the case of red imported fire ants that are well established in the Brisbane area within Queensland, there's about $400 million that have been provided for the eradication of this pest. So quite a lot of money that is going into this. And this is research that we're doing with collaborators at James Cook University. We're also looking into the possibility of detecting fungal strains in air samples. And this comes from previous research that I had done in the past with our team, where we showed that we could collect airborne samples, extract the DNA from that, and then from there, we could detect strains of something called myrtle rust. Myrtle rust is just a fungal species that affects about 170 species in Australia. And it's quite destructive. We could detect those spores in air samples, but not only that, we could also detect heavily infested tissue, medium infested tissue, and asymptomatic tissue. Asymptomatic being that they don't display obvious signs of infestation, that we're infected with the dust, with the rust. And we're using this machine that you see in the middle. It's a remote sampler for environmental samples, insects, and a couple of other environmental factors that you can just live in a location and use that to collect the samples that you need. Biosecurity officers wouldn't have to come every single day. They would just have to come like once, collect all of those samples, and they could be tested and analyzed. And that's one of the things that we want to do at the moment for myrtle rust in Australia. And I would love, if I get the chance, and if one of my new grant applications is successful, is to actually put them at ports of entry and just have them sitting and collecting these samples at the ports of entry to assess what is coming in at that stage into Australia. Well, so more pictures of when we actually tested this back in 2021. And then one of the other things that we're doing at the moment is detecting high priority diseases in Australia and overseas. And we're concentrating with foot and mouth disease, avian influenza, and lumpy skin disease. And this is an active collaboration between CSIRO and the Department of Agriculture, Fisheries and Forestry. This is happening right now, and we will see what comes out of this in about a year. And then lastly, I wanted to highlight that all of this work commonly happens through collaboration. We cannot do it on our own. And we have a wide range of facilities, of experts in both Australia and New Zealand that have created now the Southern eDNA Society, and they all are a collective that serve as the national eDNA testing facilities and programs of Australia. Now, I'm just winding down, but I wanted to just promote, if I can, if I get the chance. There's going to be a conference next year in February in wonderful Tasmania. I highly recommend encourage you all to come if you're interested in eDNA. It is a place where many of the experts of this side of the world are going to meet and share some of their results and some of the things that they do. Roger is presenting, so you can probably also come and see him present there as well. And with that, I think that's all that I had for all of you. I hope that it wasn't too informal. If you have any questions for me, I'm happy to answer them. And if you want to contact me, by all means, use those emails there, or just, how do you say? Capture the R code, I guess. That is just going to take you to a different web page where we have many of this information. So thank you so much for your time. Thank you for everyone online, and I'll take your questions, if any.
Thank you. I will probably take some questions from the room first, and then switch to Zoom. So are there any questions? Rui?
So you said you went to this place in Thailand to test their water. Like, why did they agree? Because weren't they worried you'd just make them look bad? No.
I think it was because they, first of all, I think they saw the potential of this becoming a thing in the future. It is in the best interest of international importing facilities like this one to collaborate with the stakeholders that impose quarantine measures on them. So if they fully understand what it is expecting out of them, they can make adjustments quickly, cheaply, to those biosecurity, like quarantine conditions that they need to uphold. This particular international quarantine facility is one of the biggest ones in Thailand. They were very keen on not only on the case of could we actually use this for compliance of non-permitted fish, but they were really keen on could we use this to detect parasites. I think they saw them from a perspective not of like imports and so on, but they saw it from the perspective of could we actually use this to do monitoring of our own stock, because they produce quite a lot of fish in that regard. So I think that's the reason why.
On the same idea, like the water they use for packing, you said like it's completely clean, you couldn't detect it. What do they do? Do they like filter the water? Is it like artificial seawater? What do they?
So they have in the facility 2 separate tanks, one for marine systems, one for freshwater systems. They lower down the temperature, and they also have them through high quality filtration systems. So that water is actively filtered before they actually use it for packaging.
So it's filtered water.
Yeah, correct. It's part of the requirements in of the health requirements that they need to uphold when they're importing fish internationally, not just to Australia, but to all other countries.
Okay.
That's what they do.
Thank you so much for your talk. I have a simple question. So you say that there were collaborations between Australia and New Zealand. But I wonder if you have actually spoke to the countries where these specimens came from. So what are the biosecurity methods in other countries, and is there any kind of communication between you? So when you find these specimens, do you report to them? Do you know about their methods? Can you work together?
Yeah. So yes, thank you for that. So there's 2 different components to this. Number 1 is that normally where this pest species are naturalized or well established, there's very little that you can do in terms of eradication. You can only just manage and contain what they do. So many of the countries that already have these pests well established, they have different methodologies. They either change the stock of what they would produce, something that is a little bit more resistant against that one given pest, or they just accept the quality measures of that one pest. In terms of sharing this research that we have, there's international biosecurity working committees around the world, right? Many of them concentrated on a specific topic. In the case of Capra beetle, for instance, there's the International Forestry Biosecurity Committee. I present my work to them, and then in that committee, I mean, I don't know many of the names of the people that are there, but I'm kind of hopeful that they're like the conglomerate of people that are interested in that topic. They take that information back to their own biosecurity committees. But I've never had the chance to actually go to some of the places where these pests are naturally occurring. The only instance will be varromites next year in New Zealand, where they actively have an already established infestation of varromites.
It will be interesting to see the difference between the African continent, for example, and how do they operate there as opposed to.
Yeah, absolutely. Absolutely.
Thank you so much for the interesting talk. I kind of wish you would have shown some plots from your papers, because I really can't tell how the benchmarkings were done as compared to other ways of biomonitoring. And I don't know, to me, it just seems like eDNA still, it's still hard. It's not 100% sure. It's still taking way too long. Like you said, 14 days to get back single sequencing results. So how does it compare to already existing ways of biomonitoring? Like, are the benchmarks even comparable?
I think there's many ways in which eDNA has been compared to other conventional methods. In my case, most of the testing that was done was within the context of, could this be done in the field? And within that context, I could only compare it against the benchmark methods in the lab, right? That was specifically for the purpose of assay development and so on. In the context of, for example, the 14 days that it took for testing, that's my lab, right? But there's other labs that can do this in 2 days. Like, for example, the South Australian Research and Development Institute, they have a benchmark collection and extraction method that allows them to extract and process about 500 samples a day, which is, you know, like, I cannot do that. But that's why collaborations need to be made, like, need to be essential. They need to be clear, because if I cannot do it, but someone else can, it is my responsibility to tell government they can do it better, which is what is happening in many of my projects as well. So I think there's an underlying overarching view of the limitations that we have with eDNA. But in some of these cases, one needs to be looking at those leading facilities that are pushing forward some of these technologies, and that's one of those cases. Awesome.
Any other questions from the room?
Hi, I have a technical question about the mites of bees, that parasite. So are you going to do meta barcoding for all invertebrates, or just that mite specific barcoding? Because
Yeah, sorry, I got excited. My apologies. Well, finish your question. I'm sorry.
Yeah. Because if you are going to meta barcoding all invertebrates, most of the amplification will be the bees' DNA, right? So yeah, that's the question.
Awesome. Our lab is not doing meta barcoding approaches of any other insect species, but these collaborations that we have actively ongoing include CSIRO, and CSIRO is doing meta barcoding of viruses and bacteria that may infest beehives when infested with varroa mites, for example. One of the things that varroa mite does is that they are vectors of very important viruses that infest bees, right? And one of the things that they do is that by doing meta barcoding, they can assess the diversity of those viruses at the time of infestation. And if we were to, for example, do meta barcoding on insects, by all means, yeah, I think it would be flooded by beehive DNA. There's techniques to avoid that, like you could develop a blocking primer that would block away detection of beehives, but I mean, all of that remains to be seen. I'm still very skeptical of blocking primers as effective methods to do that, but it is something that is being actively done. Yeah.
So there's not so much a question, but a comment from online. Judy Velati, thank you, Alejandro, well done. We have completed the ART additional diagnostics project here at STAFF. The time to catch up and share additional knowledge and understandings.
Okay, so thank you. This is an officer in the Department of Agriculture, Fisheries and Forestry that is actively looking at the insects and insect diversity that they find physically in the actual samples that we analyzed. So after we did the whole DNA and RNA extraction, they took those samples back and then they sieved through all of them as a, let's say, a taxonomical anchor point to some of the results that we're getting. So thank you for that. I'm keen to have that meeting.
Any other questions?
Yeah.
So are there taxa where this would be less effective? So like for reptiles, right? They're not necessarily shedding a lot or maybe just all at once. So which taxa do you think wouldn't really this be a super effective tool for?
Okay. It's interesting that you bring into collection, like into the question reptiles. Do you work with reptiles in eDNA?
Not eDNA, but reptiles.
Reptiles itself. Okay. One of my PhD students is actively working with red-eared slider turtles in Australia. And I know for a fact eDNA doesn't work that well with reptiles. He could have a turtle right there, collect a sample next to it, and have no detection whatsoever. So reptiles are one of those groups for which we don't really know that much just yet. And birds are another one of those groups for which there's a lot of essays that would target birds diversity, but the reference database for birds is, I mean, depending on the region that you're sitting, is quite low. I think quite a lot of the work that is being done at the moment with environmental DNA is in marine systems and zooplankton, bacteria and viruses, of course, in soil samples, for example. Insect diversity is one of the bigger ones and is one of the ones that we have for one of our projects. With COVID, right, there was this massive boom in the use of eDNA for the detection of diseases and how those samples would inform countries on strain level differentiation of many different diseases. So it certainly act as a catalyst for quite a lot of research, but reptiles, yeah, there's a lot in there that we don't yet know.
It's actually interesting. The Australian government was at some point interested in, we have these huge estuarine crocodiles that are quite dangerous. So there's the ponds all over Northern Australia, and they may or may not live in there. And they were interested in eDNA assay for these ponds, but no one was willing to touch it because we don't even we don't know whether we can reliably detect these crocodiles. And if we say like, oh yeah, there's no crocodiles in this pond, and people go swimming and die, so no one wants this responsibility.
Yeah.
Cool. If there's no more questions, Alejandro is still here today, tomorrow, and then Friday morning. If you have any questions, just find him or send him an email.
Okay.
Thank you.
Thank you, everyone. Thank you, everyone online. Joaquin is there. Ciao, chiquitín. Bye-bye.